Uh, Thank you for the invitation to speak. I wish next time will be in person. And I've got a lot of slides, but don't worry about it. We'll get through it quite quickly. Um, and one thing that I hope is that our rootstocks are making a difference in all your orchards and your whole industry. So I want to speak about a few things about new frontiers, new rootstocks, and things that we're working on and we're studying right now. So um, got a nice introduction. You know where we're from, upstate New York in Geneva. There's a nice lake, but we also have the largest apple collection in the world. So a few of you have come to visit. Come visit again whenever you can. The Apple Rooster Breeding Program is a collaboration among several scientists and right now, but the most important ones, and for which I am truly grateful, um, are Jim Cummings and Herb Alwinkle, and then Terrence Robinson. So they are the initiators of the breeding program. 1968, I wasn't even, I was born in 1968. So to give you an idea of how long some of these things have taken. And then Avais Khan, who has joined as replacement of Herb Alwinkle, who does some of the plant pathology work. But you can see here Terrence Robinson. Some of you have met him. Um, okay, let's see like that. Um, and some of you have met him. He is not only a talker, but he actually walks the walk. And this is a bin full of Royal Red Honeycrisp, bitter pit free. They were second leaf on G969, just perfect apples. And so he practices what we preach. And I also recognize our technicians, some of whom have passed away. So really quickly, we are a collaboration between Cornell and the USDA. I am represent the USDA part of the Geneva breeding program. And I think the introduction that was given about this symposium, rootstocks are central to uh, a lot of the orchard performance and a lot of these interactions. So with the soil and the sign variety and climate and diseases and insects and so on and so forth, and then the management style. So you need to when you design an orchard, you cannot afford to forget about the rootstocks until the very last moment. Because you get stuck with, essentially what you choose. And so we try in the Geneva Breeding Program to provide what we call designer solutions for all of these interactions. No rootstock is perfect, but some rootstocks are a lot better than others at providing value to all the apple growers. And so we've come up with some ideas about, okay, how much are these traits worth? So increasing productivity, the branch angle modification. Some of you have seen actually trees open up because of the rootstock, the replant disease tolerance, the woolly apple aphid resistance, fire blight, not so important over there, but very important over here. And then unique nutrient uptake. And I'll speak a little bit about these. And so we've released several rootstocks in the US, and I'll speak mostly about the US here, but also in South Africa. They're specific to South Africa. And these rootstocks have changed the landscape. I'm not going to speak about individual rootstocks today. This is more about different things. So these rootstocks have changed the landscape since I joined. This is 2001. It was mostly a monoculture of M9, and now this is the diversity of rootstocks that we see in the U.S. market, and I think the same thing is happening in South Africa and other parts of the world. It's necessary. It's necessary, and so here we have a table that you should be able to find online. If anybody needs it, I can send it to them. But these are most of our released rootstocks and sort of like their properties. Again, none of them are perfect, but some of them are perfect or the best for a particular situation. And by that I mean so I'm looking at just one interaction right here. So between the rootstocks and the scions, there are some things that the scions, for example, lack. And I will give a specific example of bitter pit and the potassium to calcium ratio in fruit. Lee will talk a little bit more about that, but there are specific rootstocks that will improve the potassium to calcium ratio in honeycrisp such that there is little or no bitter pit. But there are other, too much vigor, biennial bearing, and so on and so forth. So we can start thinking about that when we plant a particular variety. We need to have a particular rootstock in mind. Okay, and this diversity of rootstocks changes the way we can apply and we can build fruiting walls and multi-axis, single axis, and all sorts of different things. Rootstocks enable those things. So I'll give you some example of some rootstock-induced traits to the scion. Okay, so dwarfing and early bearing, that's old technology, and I'll show you a little bit about that. But this branch angle is fairly new. It's Geneva based or Geneva rootstock based. More flowering and bud break in low chill environments. Again, Geneva. We found that new hormone balances coming through the graft union. The mineral nutrient concentration also. We can pick high, low, and this and that in upper rootstocks. And then more traits like disease in disease resistance. So I'm going to speak a little bit about this old tech, which is dwarfing and early bearing. So this is the way things used to be 100 years ago. And this over here is the way most apple orchards should be or are now. The difference is, rootstocks. These two trees, this big tree and this small tree, were planted at the same time, okay? The only difference is the rootstock, and one is growing apples and the other one is growing wood. We don't like to grow wood. We like to grow apples, and good fruit quality apples. And so this dwarfing ability has changed the light interception, the ability to have greater fruit quality and production, and to be very, very precise in the way we do orchards in an industrial way, in a very, very efficient way, capturing light and making light much more efficient. So going from 3D to a 2D planar or something similar to that. So where did dwarfing come from? Okay, so we have a little bit of history here. You can go on Wikipedia and find find out about this scientist. His name is Ron Hatton, and he and his colleagues were the first DUS testers. They didn't come up with dwarfing, okay? I'll show you a little bit in a minute. But they were the first DUS testers of all the material, all the rootstocks that were being grown in Europe. So they got material from Germany, France, Belgium, also England, all sorts of other places. And they did very, very precise DUS testing of that material, and they came up with the Mulling series, okay? But was dwarfing around in 1866? Oh, yes, it was. How about in 1767? About 100 years earlier. Oh, yes, it was. If you could read this in French, you can see. So M9, and if it was in 1767 enough to be known in a book, that means in the 1600s it would have been present. So M9, M8, and things like that are very old technology, and we need to move on from that. I don't care if it's Geneva or whatever. If you have something better, use something better. Um, so the dwarfing and early bearing, but this is dwarfing that we're talking about. Two genes, two genomic locations. Maybe there are a few things other than that, and we can sort of figure out how to put these genes together in several combinations in order to have medium trees with lots of apples, very, very small trees with lots of apples, and large trees, and so on and so forth. But we have that we have the ability to do that to control it in the breeding program. But the what perhaps if there is one lesson that I need you to know about dwarfing and early bearing, is that at least in the US, and this is probably around the world as well, the increase due to the application of dwarfing and early bearing rootstocks, and you can see this in the change in 1982 where 40% were dwarfing, and now 93% of the rootstocks in the US are dwarfing. That change has increased the efficiency of the whole industry by about $2 billion in the US every year. Okay. That's a lot of money. That's a lot of efficiency. It's a lot of efficiency, not only for money-wise, but also environmentally. So we're growing much more efficiently, many more apples in a smaller footprint. And so when people say, apple growers are bad for climate change, and give them, tell them it's bull. Okay. This is American thing, because you have done more in applying these new technologies to save the environment and to save and to improve that than a lot of the other industries in just by applying dwarfing. Okay, enough from the bully pulpit. But apple growers have challenges in fruit production, and workers, this is are demanding less stress, and so we need to come up with ways, namely, harvesting implements and so on and so forth. Even in the nursery, it's important to have something that causes less stress, because labor is becoming a major issue, and rootstocks provide a solution, by generating these efficiencies and these new types of fruiting walls and things like that. The rootstocks are providing a solution to all of that. Perhaps to the easiness of labor, or to be able to attract better workers in your orchard if they don't get so tired. And so there are things, for example, that allow you to pick in a much more easier way. Or look at that, maybe pick, robotics that pick during the night, or platforms that allow you to apply that. Those are all things that start with having a good rootstock that can balance that tree and can make it in a certain shape. The climate, another challenge that the apple growers have. The climate is always changing, and right now the trends are showing, an increase in heating units and so on and so forth, and that is something real, especially when it affects the winter chill. And so there is a prediction worldwide in the lack of winter chill is going to happen in the next few decades. So we do have a solution for that. We found some apple rootstocks, and this is G213. It's not the only one that does that in that we found in the breeding program, but we're studying this process. as apple rootstocks can reduce actually the chilling requirement of grafted science. And this has been proven in several other experiments that we've done, even in Geneva. Um, that's all you have to know. If you need publications about this and a little bit more knowledge, just contact me. We have everything available that we can share with you. But also, there are other challenges in fruit production where consumers are demanding less pesticides, and so we have to be careful about what we do. No, we have deployed woolly apple leaf resistance in many of our rootstocks. We're the first ones that have deployed real fire blight genetic resistance as rootstocks, and that's become very important in the U.S., especially in fire blight areas. And we have deployed, I think, the very first Phytophthora Pythium combination resistance along with replant tolerance. They're connected, okay? Uh, it's not the very first Phytophthora resistance, I realize that, but about the replant is the replant package. But this is what we do is we select material that is tolerant to all those root things, and we've learned a lot about these root interactions um, with the soil microbiota. One of the things that, and again, I'm I could go for hours talking about each one of these things, but I don't want to do that today. I'm just gonna tell you a little bit about that. But one cool experiment that was done by Rachel Lyso and Mark Mazzola was to look at the percolates of rootstocks growing in sand culture, and they noticed that the metabolic, the profile was different by different rootstocks, and that metabolic profile was actually effective in changing the bacteria, fungi, oomycete, the population in the rhizosphere. Quite something amazing. And then when I looked at the bigger rootstock diversity, so blue is low, red is high levels, and then these are the compounds over here. Uh, um, okay, and so when you look at these compounds and you see caffeine, you didn't know roots could produce caffeine, but they do, or benzoic acid. And some have high levels and some have low levels. And some of these compounds are actually bioactive, okay? They have a reason for being there. So we're learning more about that and about how the rhizosphere changes because of the root metabolome. We're also, looking at the other challenge in fruit production. If you produce poor quality fruit and there is a rootstock component to that, we need more fruit, by the way, because the population is increasing and continues to increase. Maybe it'll taper off, but we need more good quality fruit for everybody to enjoy these wonderful things. I'm a fruit guy. I just love fruit. And so, how do rootstocks affect fruit? Have you ever thought about that? Um, and it turns out that there are changes in tree architecture that may change the sun exposure, but water availability, the nutrients, and so on and so forth. All of those can change the fruit quality, and in such a way that is quite significant. You may have seen this slide before, but we've repeated the same experiment with several different other types. This was the same gala, and color, firmness, maturity, sugar, size are affected by apple rootstocks. So an apple grown on one rootstock is different than an apple grown on another rootstock when you put it in storage. So just know about that, okay? And one of the ways that we have found that, and Lee Calcis will be talking more about this. One of the ways that we found is that predictably, this is a group of, this is boron, for example, the boron levels throughout the years. So measured through many years and different things. And you can see M9 and something similar, very low in boron absorption. The best of boron absorption right in this group right here, where G935, 41, and some of the other ones are at, and 484. But this is similar in phosphorus, potassium, and some of the other nutrients. Calcium is a little bit more complicated, but regardless, that has an influence on fruit production, okay? And also the absorption of nitrogen. This is just, I don't want you to remember this, but just two major genes affecting nitrogen absorption, okay? And that we found, and you can see in the sibs of G213 and G214, okay? 214 has the low nitrogen, and 213 has the high nitrogen, okay? And you can see, they're full sibs. Pretty interesting how you see it pan out in a huge nursery like that. But that nitrogen and also the potassium and everything else, they affect fruit quality. This is bitter pit that we're looking at, and we measure bitter pit with an X-ray machine. Well, we don't measure bitter pit with an X-ray. We measure the potassium to calcium ratio with this X-ray machine. Each one of these dots is a different rootstock, okay? And these are 10 apples from 5 trees replications. So this is a very, very good number. And you can see this a very strong correlation between this potassium calcium ratio and the bitter pit presence. But that is money. That is money in for apple growers, because if you have like 40% bitter pit in your crop because you chose you picked M7 as your rootstock. Sorry, that's a weed. You pick M7 as your rootstock and you're getting very poor things, or you pick another rootstock that has high bitter pit, then you're losing money. And sometimes, over the life of the orchard, over 10 years, you can lose close to per hectare, close to $250,000 just from culls, okay? And how do we know that? Well, we know that because we've done decade level research with our rootstocks, and it's not just us, collaborators from the NC 140 and all sorts of other scientific friends. But you need that with rootstock. You need to have long-term knowledge or long-term performance knowledge. And so many of our papers that we produced lately have this moniker, long-term, because a rootstock stay, these trees stay for a long time. Something else that we're studying, and then one of the challenges that we're trying to resolve, is water availability and quality, okay? Less water may become available, or the quality of the water is going to be pretty bad. And we're using aeroponic systems to study apple roots, okay? This is a very interesting systems where a mist of nutrients gets applied to the root system. They grow in air, okay? And you can apply some very, very precise treatments. This is a scheme of how the system works. I don't need to tell you much about it other than it exists. It was developed by NASA a long time ago, and we use it. But we use it to study mineral nutrient absorption, the pH associations, temperature, root pathogen, salinity, and drought. And we have easy access to the root system for RNA and for all sorts of other things. That allows us to get a nitty-gritty knowledge about how the root systems of our rootstocks work. And so this is an example of a paper that we just published with the relationship between pH, low or high pH, and some of our rootstocks, okay? And again, pH is a very important consideration when planting an orchard because it affects the way the rootstock performs at the basic level of nutrient absorption, but also how the roots grow. They seem to have that ability. But we can also study, this is a large format tank, and I built these dendrometer sensors to study the relationship between evapotranspiration, or we can give some very, very good drought treatments with these large format aeroponic tank. This is Davis Upchurch, our student right now. And this is inside. You can see the roots just hanging out, and we can study the roots, while both the root system and the scion as they grow. And you can see that some of these dendrometers, or they respond very, very finely at the amount of light that the trees are receiving, and you can see how fast they respond. So this is the light day and night, and cloudy day, partially cloudy. And you can see how the tree sort of shrinks during the day, and then during the night it swells. And then when it's cloudy, it doesn't shrink as much. But then when it's partially cloudy, you see how fast it's reacting to the sunlight and to the to the clouds. So we're trying to study different rootstocks, and you can see that different rootstocks are behaving a different way. Okay, so this is, and you can expand this to drought, and we can add the temperature to it. And this is a drought event that we applied. Again, this is that same drought event, and another drought event, and a little bit of that. But you can see how the rootstocks are behaving in a different way according to their genotype. There are other challenges. I've got 3 more minutes. I got other challenges in fruit and nursery. They're getting much more expensive to produce. And so we have tested, the use of micropropagated versus conventional propagation. In essence, they behave the same. Our rootstocks so far have behaved the same coming from micropropagation and versus conventional. In fact, sometimes they perform in a better way because the number of primary roots is increased in propagation done by micropropagation of soft cuttings, okay, when compared to layer bed cuttings. I'm not kicking away layer beds, but what I'm saying is that there are some applications, like, for example, being able to plant in the middle of summer, with a nice root ball system that will stay alive as long as you give them water right away. But yeah, you can have a lot more flexibilities with that. And then for our program, micropropagation has become very, very important. So it's become very important to release new apple rootstocks. We can bump up to 1 million rootstocks in quite in 2 years versus 5+ years when the other methods were employed. And so we have a lot of nurseries that have incorporated this into their sort of growing. These are standard and tissue culture material, directly Willow Drive. This is another nursery in California, Sierra Gold. And again, containerized trees, people testing those because they are easier to transplant. Again, another more tissue culture and containerized trees. And then we have had entire Geneva rootstock field days for several years in Brazil, in Mexico, and I hope to see you in a field day based on Geneva rootstock maybe next year we can think about that. But I thank you for listening to me. And rootstock breeding is what we do. We hope that we're making a difference in your country and in your industry. And I'm grateful for this opportunity. Thank you.